Our next presenters are from Andelyn Biosciences. They are Andrew Moreo, Head of Process Development and Preclinical Production, and Samir Acharya, Associate Director for Process Development. Welcome, gentlemen.
Thank you. So, thanks, Perry, and thanks for giving us a great forum to share what we're doing here at Andelyn. Want to talk to you today a little bit about Andelyn, give you an introduction into our capabilities, then talk about our Curator platform, give an overview of that, and then some of the updated data that I think most people will be most interested in, and I'll let Samir Acharya do that. And then if there's time, we'll talk about how optimization impacts capacity and cost of goods. Starting off, when we spun out of Nationwide Children's Hospital in 2020, what we were really looking to do is leverage our depth of knowledge in AAV manufacturing from a research and clinical phase perspective, and build out a commercial capability that really allows us to be a concept to commercial gene therapy CDMO. And I think we've really done a great job of doing that by having a complete end-to-end capability from plasmids, process development, vector manufacturing, all the way through fill finish. That includes, any analytical development and quality control, full quality oversight, regulatory support, with flexible partnerships. And flexibility is going to be a theme in this talk, as you'll see. We have 3 facilities. We have the Andelyn Development Center. That's where Samir and I call home. It's 42,000 square foot, and it's dedicated to preclinical manufacturing and process development, as well as analytical development. And then our corporate center is 180,000 square feet. It's dedicated to GMP and commercial manufacturing. We can also support toxicology manufacturing there as well. It has, it has full fill finish, quality oversight, supply chain and warehouse capabilities as well. And then our plasmid facility is where we produce all of our research tox and GMP materials. It's a 9,500 square foot facility that is our legacy facility from AAV manufacturing on NCH's campus that we retooled for plasmid manufacturing. We also do cloning services and have helper backbones for clients that need them. We tried to outfit those facilities with equipment that allowed for ultimate flexibility and scalability, regardless of the modality of manufacturing on the upstream side or the method on the downstream side. Again, we're talking about flexibility here, all the way from materials through quality control testing and fill and finish. So, that brings me to platforms. And, I think a lot of us have seen the recent draft FDA guidance on platform designations, and a lot of us CDMOs are looking at how to leverage that, as well as some of the recent comments from the FDA around platforms. And some of the speakers earlier did a great job of discussing the power of a platform and what it really brings to the table in terms of consistency of product, consistency of analytics, as well as standardization of costs and goods and streamlining of manufacturing. But there's also some holes in that that really need to be taken into consideration. And based on what the FDA is said and what we've seen as a CDMO, we realize that some of these holes are around key program factors like indication, patient population, delivery route, all of these things lend themselves to different scales, different CQAs that we really need to leverage in our platforms, as well as optimization in our platforms based on serotype, transgene, and those combinations that give us a predictable outcome, in terms of scale down models, feasibility of speed, and ultimately what that impact will be, and the cost per patient, and how that lends itself to key quality attributes in terms of the scale and in titer formulation and quality attributes as well. So, how do we address this? So the way we approach it here at Andelyn is we really have taken the word platform and its etymological roots of, which literally is a ground plan if you look it up, and defined it as such, where this is a place to start. It's a place to build from. We don't have a plug and play approach in terms of trying to pound a square peg into a round hole. Rather, we take a modular plug and play approach where we can adapt different serotypes, transgenes, and take them through and plug in optimized systems, whether that's on the upstream, the modality of manufacturing being suspension, adherent, an HSV system versus baculo, plasmid ratios and transfection cell densities we can optimize based on that, and we've decoupled the downstream or uncoupled the downstream, so we can marry any upstream process to any downstream process depending on what the client's needs are and what the product dictates in terms of quality attributes and performance attributes. So, anything from, all the way from cell expansion through final fill, there's multiple ways we can tweak, optimize, and fine tune the process to get the optimal system for any given AAV gene therapy product. So, big part of that is our cell line. We have our AAV Curator cell line that goes with our Curator platform process. We take it where we can manufacture in any cell line. This one is optimized through DOE to systematically improve AAV titers for multiple serotypes. We've assessed productivity as well as other quality attributes such as empty full. And we've optimized conditions to maximize vector production. The idea being able to grow at a maximum cell density, but transfect at a minimum DNA quantity to get the maximum and best quality product at the end of the day. So, to summarize, the Curator platform, we've tried to build something that's ultimately configurable. It's a modular platform with assessment and strategy adapted to each program that drives faster optimization. We tried to build in optimization by design using quality by design attributes, guidelines to drive our DOEs, with lean stepwise focus on process understanding and maximizing program efficiency. And then everything is ultimately data driven. We're able to leverage 15 years of manufacturing data in order to achieve high yields, high quality, and predictable performance. So, speaking of data, at this point I will turn it over to Samir to talk about some of the data behind this platform. So, Samir, it's all yours.
Yeah, thank you, Andy. Andy mentioned there's a lot of optimization that goes behind establishing a platform. And we've done that with the different cell lines. starting from small scale flask productions to 2,000 liter productions. So since we have made our own cell line, we are using the same approach to optimize this cell line for maximizing productivity as well as quality. This slide just summarizes a whole host of factors that we consider, not just for optimizing the upstream part of the platform, which is factors 1 to 5 listed here. And that includes cellular factors like which cell line we are using, what cell density to use, and so on, going to media factors, transfection reagents, which are important. How do we complex the transfection reagent with the DNA? What is the complexation volume? And going back in the end to when do we harvest our cells so that we maintain both the amount of titer we are getting as well as the quality of the product that we're getting out in terms of, let's say, what Andy mentioned was full to empty ratios. And similarly, we do downstream, what filters to use, what chromatography chemistries to use, buffers, and so on. An example of the outcome of such studies is given on the right. The top right is a contour plot. And this is an example of how we assess the effect of complexation volume, transfection reagent complexation volume, and the incubation time of that complexation on the titer. And the red region gives us then the ranges that we can use. And these ranges for all these factors then establish our process parameters. And some of these ranges then become critical process parameters because we are able to understand how, with different scales, how these factors play a role and how important they are. And one of the things that we monitor as we are developing these are based on the assays that we have is shown below, which is in this red region, how does the transfection complex look like? And here we show with dynamic light scattering, there is a uniformity of size. We have found that such an assessment is very critical because this uniformity of size is what makes that factor important for maintaining consistency in titers. So having said all that, as Andy mentioned, we've created our own cell line. The next slide then shows what we did. So we have been using our adherent curator cell line for many years, and we converted that cell line into a suspension cell line. So we derived suspension cell lines from this. And once we had those cell lines, we used the WIPS system to isolate single cell clones. And once we had a single cell clone cell lines, then we evaluated them for productivity and compared them with the commercial cell line. So mind you, we have established a set of conditions, optimized set of conditions to maximize productivity for a commercial cell line. Using those conditions, we then said, we use that to go further with our clones. So what is shown here are just 6 clones that we got, which were giving either similar or slightly better productivity. So the clone in yellow was chosen. We made a master cell bank from this. And then we used that for optimization using the same set of conditions or strategy that we mentioned earlier. The outcome of that is what Andy showed earlier, is this. So once we had an optimized set of conditions for our cell line, we compared that with the commercial cell line. As you can see here, for different serotypes, for many serotypes, our cell line performs better. For some serotypes, like serotype E, is similar. But the point here is that we have adaptability in our system, and Andy mentioned flexibility. So we have an optimization toolkit, and we can use that for any cell line. So with our curator suspension cell line, having done that, and mind you, this optimization is done with the GFP as a gene of interest, but clearly the GOI plays a role in optimizing client product. So the next slide then shows a real-time case study. Since we have a standard set of conditions here, we don't have to re-optimize everything. So for this case study with serotype D, we chose cell transfection density, cell total DNA per cell, and plasmid ratio as the factors. And we try to understand what is the effect of that on productivity and full capsids. So the top panel on the right shows a contour plot where the color indicates productivity. Red is the maximum and blue is the minimum. So from such contour plots, we can understand what ranges of factor A and B and C give us maximum productivity. On the bottom panel is what ranges of same factors give us maximum full to empty ratios. So analyzing such contour plot, we can come up with a balance between productivity and quality of the plasmids. So for this client, we went ahead and got a set of conditions for maximizing titer as well as full capsids. And the next slide then shows you what we did with that. So that we then scaled it up for 2 liters. The slide also shows you a similar study that we did with another serotype, another GOI. And as we can see that there is consistency and scalability, in this case, up to 2 liters. Now, we have established scalability with a different cell line for 2,000 liters. We're doing the same with this, with different clients as we go from 2 liters to 50 liters to commercial production. So, as Andy mentioned, this is as far as upstream is concerned. As Andy mentioned, we have modularity in our platform. So we have a variety of systems optimized for purifying the viruses. It can involve ultracentrifugation. Ultracentrifugation can involve adexanol or cesium chloride. We also have all column methods, and clearly for each GOI, each serotype, the column chemistries will differ. But what is shown here is our studies with a cell line which shows the consistency in product downstream recoveries and final purities, whether we have an ultracentrifugation method or an all-column method. In the bottom panel, we show an example of whether we use PEI or factor V as a transfection reagent, whether we use a 50-liter or 200-liter bioreactor. We have seen consistency in the quality and also the final recoveries of this product. So on the whole, since our platform is modular, we can mix and match different modules. Each of these arrows here show clarification is a module. We can use upstream and use a different clarification and a different column purification strategy over here. So that is what we are doing with different clients. And as and when how the factors dictate how the product performs, we can change that. So this is a strategy we use for many of our clients. And so what is the impact of all this in the overall scheme of things? So I will send it back to Andy, who will then discuss what is the impact of this on the different productions. Andy?
Thanks, Samir. Yeah, this is an overly simple representation, I think, but it really does illustrate what we're talking about in terms of impact of optimization in our Curator platform. So, you take a scenario of a final target yield of 1 to the 17, a very average manufacturing campaign with 500-liter bioreactor, and plug in a transgene and serotype that's with no optimization. It's something we can absolutely do. You can absolutely achieve it. If it has an average yield of 2 to the 14 VG per liter, we can do it in 5 upstream manufacturing sublots with 2 downstream purifications, certain pooling strategies, pool everything together for a final formulation of 1 to the 17. Absolutely achievable. It takes 10 weeks to do. And I won't even say having a highly optimized process, but just an optimized process. I'll stop short of highly and say just an optimized process, where if we can double the upstream manufacturing yield, which is a fairly modest improvement from 2 to the 14 VGs up to 5 to the 14 VGs, and we get the same 20% recovery, we've now reduced that processing time in suite to 5 weeks, which is a 50% reduction in processing time, a 60% reduction in cost of goods. But then if we are able to do the downstream, optimize the downstream, double the recovery from 20% to 40%, which is something that is I won't say overlooked in the industry, but because I think it's something that is actually focused on quite a bit, is the fact that we lose 80, on average up to 80% of the vector we manufacture in our downstream purification. So focusing on increasing that recovery percent even twofold to 40% recovery takes that whole process down to 3 weeks, one upstream, one downstream, and into final formulation, which is overall a 70% reduction in processing time, an 80% reduction in cost of goods, not to mention a higher quality product at the end of the day with better empty full enrichment, better purity, and better consistency in overall recovery. So, like I said, it's very simple math, but I think it really illustrates the impact of having a platform that is optimizable by design, that is modular, and can be fit to a specific product. So, with that, I will say thank you again and ask if there's any questions.
Well, thank you, gentlemen. We have time for one question, I think. So maybe to you, Andrew, because I think you brought this up. Can you talk a little bit about why plug and play doesn't work for AAV-mediated gene therapy, and what are the associated risks?
Sure. Plug and play like we were saying earlier, it can be dangerous because you are there's a lot of factors to consider in terms of scale, as well as manufacturability of the vector, performance of the vector, how we have to optimize it. For instance, also throughput time, right? If somebody has a small indication, say ocular, an adherent system may be perfectly suitable, and also it's by the way also commercializable. As opposed to a systemic delivery that has, that is more of a prevalent indication where, scale becomes a factor that affects all the upstream. It all affects based on that upstream, it affects the downstream modalities that we have to leverage to get to suit the client's needs and ultimately the patient's needs at the end of the day.
Okay, and very quickly, I'm going to slide another question in. One of the audience members is asking about capacity. Do you have current capacity? Any wait times?
We do, and I would say please contact us and talk to our business development team about that. We do have capacity. We are booking capacity now, actually, into next year. So, yes, anybody's interested, we would love to talk to them, and they can contact our business development team to do so.
There are other audience questions. We're going to have to move on, but perhaps you can follow up with them offline. Somebody has very keen interest in slide 9, wants to go over it in more detail. So thank you very much, gentlemen. Samir Acharya and Andrew Moreo from Andelyn Biosciences. Thank you for joining us.
Thank you.
Thank you.